a real pleasure to have this opportunity to sort of reflect on well over 10 years, 13 years of being director of the BRC, which was a new event. We were the first BRC in Yorkshire and were the only BRC in Yorkshire for much of the time. It was announced, the NIHR was created out of money taken, which was originally given to teaching hospitals, and Gordon Brown was the instigator of bringing this money back in, and then we had to apply for it, and then it was allowed to be brought back for research for the various institutions. But it was a competitive bid, and as I said, we were initially the only one. And now we've been in existence for 3 iterations. I'm delighted handing over to Philip, who will tell you about what's happening in the new BRC. And what we're going to cover is what the previous BRCs have managed to achieve, what plans we have for the future of the BRC, and Amy's going to be talking about PPIE and how it will relate to the new BRC. And for your engagement, the questions can be submitted to Amy and can be done through the chat channel now. If there are any questions at the end of my presentation that people would like answered straight away, I'm very happy to do that, because there will be a moment or two when Philip takes over the screen for his presentation. So this is the way it worked previously. I'm just going to move the pictures, which have got slightly in the way of the... Our research was undertaken through in the final BRC, 2 research themes and 7 work streams. So we had the 2 themes listed here: optimal treatment and preventing disability and immune-mediated inflammatory diseases. And I think most of you know that it's abbreviated to IMID. What this means, these are the active inflammatory diseases that if you don't treat, get much worse. Philip will tell you more about osteoarthritis and its pathogenesis. But I think the biggest achievement we made through those years of the BRC is that we've developed what I'm happy to say is probably the world-leading centre in autoinflammatory and immune-mediated disease. And if I list them here, Kalvia Mankia and myself ran the European guidelines for pre-RA, defining them, and has been and Kalvia has been managing now in collaboration with Oxford, Birmingham and others, our pre-RA program. In scleroderma, Francesco Delgado, who's become professor. Francesco is now head of the European Eurostar consortia, which runs scleroderma in Europe. In lupus, Ed Vital has been leading the understanding of interferon. He's a senior member of BILAG, which is a British Isles lupus group that does assessments, which I was originally involved with in Birmingham, and is now a national leader in lupus. Dennis McGonigal, Helena Mazu-Ortega, both internationally renowned in psoriatic arthritis, and I'll come back to that. Sanisha Savage, who's also becoming a professor, who has been awarded a £2.5 million grant from Oxford to study autoinflammatory disease. In polymyalgia, Sarah Mackey has run and led the guidelines for polymyalgia for the British Society of Rheumatology. And in giant cell arthritis, Anne Morgan has been leading a number of the consortia looking at the genetics. So what we have is the most complete coverage of autoimmune disease with world leaders in each area. And it's a source of great, pride, and also that we've managed to get in Leeds a unit that can hold its head to any in the world and has been officially rated #1 in the world. So the second thing we achieved was in the area of prevention. If you're going to prevent disease, you have to identify patients in the pre-disease category. And we've done this in a number of areas. The pre-RA was the initial model. In scleroderma, we've got a big cohort of patients with Raynaud's phenomenon, which is the first vascular sign of scleroderma before it becomes a fibrotic skin disease. In lupus, there's the autoantibody, anti-nuclear antibody, and we have cohorts of that, I'll show you. In psoriasis, psoriatic arthritis is preceded in 70% of cases by psoriasis. And if you can identify which patients with psoriasis are going to develop the arthritis, there's a possibility of preventing it. And all of these have this enormous value because all our diseases, once established, are permanent. If you can prevent a single case, that saves hundreds of thousands of pounds to the NHS. So if you have quite complicated ways of diagnosing patients and predicting what they're going to do, it's still cost effective to use it. And on top of these pre-disease cohorts, we have world-leading cohorts in autoinflammatory disease, polymyalgia, transcell arthritis. And as I said before, it is the most complete coverage of autoimmune disease that there is, not only in the UK, but in the world. So we're very, very proud of these groups, and I think they're in a position to go even further. I'm now going to take you through a little bit about what happens in rheumatoid, which has been the model for prevention. And I'm having great difficulty in getting everyone's pictures off my slides, but I will. If I can't read all my slides, that'll be too bad. But this is the, for some time, we've been aware in inflammatory arthritis and rheumatoid arthritis in particular, that things start with a genetic risk, but the genetic risk alone doesn't make you get the disease. But if you have environmental risks, which can include smoking or actually being a teetotaler is a risk factor, you then get local autoantibodies at the mucosa. And I'm not going to say anything about that, but we've got an extensive program with the dentists looking, and many of you may have been through the program of preventing periodontal disease, preventing autoantibodies at that site, which then go into the blood. And then just before you get disease, there's an expansion of these autoantibodies, and then subsequently the progression to rheumatoid is probably inevitable. But when we started, we didn't even know this, but when we established that these antibodies did increase, we didn't know which ones. And there was a major advance when we collaborated with colleagues in California for the first time, looking at the combination of antibodies in patients at this particular time of disease. And you might think this is pretty straightforward, but it's been very difficult. It's taken 15 years to collect the correct cohort of patients. And this is what we found. Previously, it was believed that CCP titers, either low was a low prediction, high was a high prediction. But what we found, if we combined them with CCP, low, and it was CCP negative, dropped from 7% likelihood of rheumatoid arthritis to under 3%. If it was high titer and CCP was negative, the risk for CCP2 went down to 9%, a four-fold reduction. In high titer, if it was positive, it went up to 45%. So we have a five-fold difference in the same patients who would previously have been categorized at high risk, depending on what the other antibody showed. This was the first time this had ever been discovered. And so it shows that in patients who have this antibody, which is the best described risk for developing rheumatoid arthritis, having this other autoantibody provided valuable additional information, increasing the risk, the predictive risk of the original antibody by 5, going from 9% to 45%. And to show you what that means, in low titer, these antibodies, as I've said, went from 7.5 to 3.3. High titer went down from 39 to 9. So you could either have low titer or high titer, but depending on what the other antibody showed, you had a very under 10% risk, which means you could probably be reassured. Conversely, if you had low titer, we would previously have not worried too much about you, but it goes up to 25% when you have the other antibody, which there's a poor concordance between these two antibodies. And it goes even higher if the CCP2 was high combined with the CCP3. So high CCP3 allows us close follow-up and trials on prevention. So this was a big advance that we found, which is a major improvement, because it does allow us to actually stratify patients for follow-up in our pre-RA clinic. But the other thing that we found, and I'm not going to, there are unfortunately a huge amount of data here, and I've only selected a small amount, but the patients who have, before they have any clinical arthritis, if they have subclinical disease in their joints, that is picked up by MRI or ultrasound, they have a high risk of progression. But one thing we believe, and we have found, is that ultrasound inflammation is a late feature and may already indicate you're destined to get rheumatoid arthritis and may not be in the preventable stage. And it's articulated here. Is it too late to prevent rheumatoid arthritis if joint inflammation, ultrasound, has occurred? In other words, we needed to know, how the antibodies related to subclinical arthritis, because the likely thought at that time was that as soon as the disease gets into your joints, it changes your immune status, and then you produce lots of antibodies. But we hoped that what we could find was that the antibodies preceded it, because if that were the case, you could find patients with the antibodies who wouldn't actually have any joint involvement, and the standard treatment of stabilizing things at that point would mean those patients would never get joint disease. So the question was, did the ultrasound abnormalities precede or follow the autoantibody changes that I described before? So to do this, we had to find patients who didn't have ultrasound at the beginning. And first of all, we looked at about 100 patients to see if they all went through a phase of subclinical, which means imaging arthritis. And over three quarters of them did. Some of the others we weren't able to do because we didn't have the right times for the antibody. And then when subsequently looked at these patients who didn't have any ultrasound but did develop it, were there antibodies there before? And the answer is, it's not easy to tell from these graphs, and I fully accept that, but the answer is that these patients actually developed the changes in their antibodies before they developed the changes in their joints, which means it's fantastically fortuitous, because we can monitor with a blood test, and we can pick up patients before the inevitable joint disease occurs. And this is what it's enabled us to do. So we have the genetic risk, the local autoantibodies in the mouth, in the gut. They then get non-specific symptoms, at which point we do their antibodies, and this is where that extended antibody profile is relevant. And only subsequently do most patients get the ultrasound abnormality, and we have the gap between going from here to here as what is now the true window of opportunity. And in fact, we also know in other work we've done that the first joint abnormal, or musculoskeletal abnormality, actually isn't in the joint, it's in the tendons and in the around the muscles of the hand. These are the first structures that are affected before the joint. So this has given us real hope that we can actually intervene before joint involvement and make a long-term difference. And now with these sort of informations, the studies are coming on stream, and we're starting a new study literally next week. And 2 other studies have shown it is possible that you can prevent, at least for a time being, the development of rheumatoid arthritis, dependent on this sort of model of what is occurring. So this summarizes it. The extended autoantibody profile, which we now measure in individuals with symptoms, musculoskeletal symptoms, which precedes joint involvements, in whom subclinical joint inflammation is not present but imminent because they have this, the antibodies, are probably the ideal target population for therapeutic trials for arthritis prevention. And this is something I've personally been trying to pursue for the last 40 years, and to have now biomarkers of prediction and the ability to get in before the joint damages occur, I think is a landmark in the understanding of rheumatoid arthritis. Now I'm going to show you, moving on to scleroderma, systemic sclerosis. This is a very complicated slide from some very sophisticated scientists. And you can't really tell, I'm sure, what's going on here, but this is Raynaud's phenomenon. And this starts off with anti-centromere antibody, which is what they use as their marker of pre-disease. They then develop further antibodies, topoisomerase antibodies, and then they have skin involvement, interstitial lung involvement, pulmonary hypertension, and they are doing the same as we've done in rheumatoid, and identifying biomarkers which predict which patients go on to get progressive skin disease, progressive interstitial lung disease, and increased pulmonary artery pressures. And they're doing this with a variety of different organizations, Chemomab, Boehringer Ingelheim, particularly interested in the vascular disease, AbbVie, the translation study to identify mechanisms of progression of disease in patients at risk, and AstraZeneca, who are particularly interested in the interferon. And it does seem a common feature to all these diseases is that they have a period with autoimmunity, which is an autoantibody, and increased interferon. So scleroderma, it's catching up rapidly. It was a disease with very little science behind it. It's one of the great expanding areas and is ideally suited to prevention again. Lupus, Francesco is running scleroderma, Ed is running lupus, and they've been doing the same thing. This is taking patients with autoimmunity, anti-nuclear antibodies, and that what we've shown in these longitudinal studies, 3-year studies, that there are some patients who develop lupus, other patients, the vast majority of the antibodies, actually have either no disease or a disease different to lupus. Now, one of the things we're very interested in is actually trying to help the patients in this category because they're not well. They don't have lupus, but they're not well. And understanding the pathogenesis, I hope, will make a bigger difference to this group almost than to lupus. But we do have now biomarkers of which patients, the smallish 1 in 10 of patients who have ANA who will progress to lupus. And one of the big drivers of this, again, is interferon. And you may be aware there are specific monoclonal antibodies that block interferon, which are very attractive to use in this phase of disease. Another thing that we've been looking at is actually the interferon in skin, and showing that it's actually largely in skin keratinocytes that the abnormalities of lupus occur. And this is patients who don't have lupus, don't have skin disease that you can see, but they're at risk on the basis of their antibodies and their interferon. And you can see that there's a marked difference between these at risk and normal, and that's reflected by the high interferon. And this is a bit of a cheat because we've got a break in the vertical axis, but this is lesional lupus producing lots of interferon. This at-risk population also produces a lot of interferon. So these patients who don't have the disease have all the pathology occurring beforehand, and then it's just waiting to be triggered to become clinical disease. Another study that we did with Yusuf, who was to give local injections of anti-TNF, which blocks the cytokine TNF in 1 milligrams amount. It was being given at 50 milligrams systemically. And although that works on the local skin lesions, it does make the general lupus worse, which can give you kidney disease. So it's dangerous to give the blockade systemically, but we actually rationalized that it was going to be safer, more effective, and more cost-effective to inject it actually into the lesion. And this was a very successful project. This shows discoid lupus is one of the hardest things to treat. And we got a 50% improvement in about half the patients we injected it in. So it was a very successful study, and it's given a proof of concept to a completely different approach for a very difficult aspect of lupus. One final thing with lupus was the understanding of the joint disease of lupus, which can be, it's present in up to, well, just over 90% of patients and can be the dominant symptom. And these we looked at 112 consecutive patients and using our imaging capacity here. What we showed was that ultrasound-only synovitis was associated with worse symptoms and serological markers. Ultrasound-only meaning that you couldn't see any joint disease. And this is what you find, that about a third have no confirmed synovitis, just under a third have ultrasound synovitis, and just over a third have clinical synovitis. So it's almost equal proportions of clinically relevant joint disease. Joint disease is only seen on ultrasound, but is markedly symptomatic, and then nothing at all. And this, of course, is going to help us manage lupus, because some of the lupus is very like rheumatoid. Others is not like it at all. It's capsular and has some features perhaps more in common with mechanical joint disease, and they do need quite different treatments. In psoriatic arthritis, as I said, we've been looking at pre-disease. This is a study done by Laura Savage with Dennis McGonigal. And basically, these are patients from the psoriasis clinic without clinical arthritis, but have, they fill in a questionnaire. If they have any symptoms, they have imaging. And this is showing a chronic thickening of a tendon. And what they showed was intervention with particular therapy had a marked effect on inflammation in patients who didn't actually have overt disease. So the principle here is exactly the same. It's the hope that treating patients before they've got either enthesitis, which is a tendonitis, such as Achilles tendonitis, before it occurs, will prevent patients transitioning to psoriatic arthritis. Other things they've done, they've helped explain the spinal disease in spondyloarthritis, ankylosing spondylitis, of which there is a form in psoriatic arthritis, but it has explained why certain forms of therapy don't work in the spine, but do work in the periphery, which is a very fundamental finding. And some of their other dermatorheumatology successes have been, they now have regular, many times a week, dermatorheumatology clinics. They screen PSA in the community. They're actually at the Merion Center this week with the dermatologists picking up new cases of psoriasis. And there are 10 consultants working in this area with national awards for the service for Helena. And they've got academics now moving into this area, which has always been a stronghold in Leeds, and it's fantastic to see it's really going far. And Philip Helliwell, who's been here for many years, working in Bradford, is leading in psoriatic arthritis metrology, which is the measurement. And finally, one of our other groups, which is stratification, it's a complicated slide. I won't go into too much, but it was an attempt to look at what happens if we don't treat patients perfectly, and if we can get round it. What we found, this was patients who were randomized to receive a TNF blocking agent with methotrexate, or to start off with standard therapy, and then add in this if they did not get remission. And it showed that the expression of genes changes, and it's more difficult to get remission if you're 6 months later, but it did together show that we were able to get large numbers of patients into remission, and there is an advantage of targeting patients at the start. And we are now nearly finishing a study which we've been doing, which has approximately 100 patients in it, where we actually do a test at the beginning of their arthritis to show whether they'll respond to methotrexate or not. And if they don't have the, if we show that they're unlikely to respond to methotrexate with remission, they actually receive early TNF blockers as a part of a randomized study. and that will be yielding its results in the next few months. Finally, the thing that we've been obviously for the last couple of years interested in and involved in is vaccination, and I spoke about that with the people who have been most involved, which are Benazir Salim and Youssef, at a last ask of the researcher a month or so ago. And what we were able to show was a defective antibody response, especially in patients on rituximab. But fortunately, the other side of the immune response, the T cell response, especially in rituximab, was much less affected. So things weren't as bad as they apparently appeared. And we've just had a paper accepted by Lancet Rheumatology illustrating that the rituximab in our hands, in our clinic, didn't do any worse than they would have done outside COVID. And I think that's a credit to the patients who were very good at getting their vaccinations at the right time and shielding when we told them to. But there were no excess deaths in our rituximab population at all, which has not been the case elsewhere. We also showed that there were differences between the two vaccines, and the AstraZeneca was particularly good at giving T-cell responses, which are more long-lasting, and that was used to explain some of the reduced deaths in the elderly in this country. Although overall, of course, we didn't do brilliantly as a country. We showed that there was a 10% reduction of serious infection every time you were vaccinated, and really encouraged all our patients to have the extra booster vaccines as soon as they become available. As I said, rituximab is complex, but we have managed, I think, to find a way of vaccinating effectively, and the key to this is to seeing patients regularly and making sure they're vaccinated before they have their rituximab, their next dose of rituximab. As we pointed out, it would be ideal if we could have managed the vaccination ourselves appropriately with the therapy, because that would have been, of course, ideal.
I'll now talk a bit about the non-inflammatory or immune-based arthritises that we deal with, and this side of the work is quite a lot of patient numbers. Again, what we see here is that this has involved a lot of people, both in the osteoarthritis group. You will have seen Dennis, who works across groups, across inflammatory arthritis and in osteoarthritis, in joint replacements, and on the technology side, the bioengineers, of which John Fisher was a leading person in the UK, who's retired just during the course of this last BRC, along with his partner Eileen Ingham, who was not only a bioengineer but a biologist who helped develop some of the scaffold products. These are the collagen and structural infrastructure of your joint that you can decellularize. You can take the cells out of, say, a pig bit of cartilage, remove all the cells, and you're left with a scaffold. And then you can implant that scaffold in humans, and they repopulate it with their own cells. And Eileen led a group that had done some of that work. The biggest advances of that work so far have not been in MSK diseases, but in cardiovascular disease. And Ruth Wilcox, another bioengineer, has now taken over from John and Eileen in running our Institute of Medical and Biomechanical Engineering. So they work closely with us, and with Hemant Pandit, who's the Deputy Director of LERM, our Arthritis Institute, and an orthopaedic surgeon who's very active both in OA and in joint replacements. And of course, all these joint problems like osteoarthritis, tendon problems, are all related to what we do and how we use our body. So biomechanics are very critical in understanding. So these three overlapping groups of people work closely together. It's worth pointing out that this problem is not going anywhere at present. It's really increasing a lot. Between 1990 and 2019, there was a 113% increase in osteoarthritis reported in the world. The numbers are just massive. We're talking hundreds of millions of people, in part due to aging and in part due to obesity, all of which is increasing not just in the Western world, but across a lot of the world. So joint replacements are important, but a lot of the stuff, because it's hard to do trials in joint replacements, because your average joint replacement lasts 20 years. Nobody's going to do a randomized trial for 20 years in somebody. So a lot of the work that is done in joint replacements, improving their design, is done in laboratories, and it's done on computers, trying to model how do we make things, how do we make them work a bit better. We know people are living longer, and we know that people expect to get really good joint replacements. Not everybody's going to have an Andy Murray who can go back to playing elite tennis after having a hip joint done. Most people are happy if they get reduced pain and average function, but even those expectations are growing. And the bioengineering people do a lot of work in these simulators that you can see on the left here of this slide, which are basically putting repeated loading through a joint over a long period of time and increasing the number of cycles and the amount of wear and tear that comes out of these joints. And they do computer modeling, such as on the right-hand side here, where they say, after I've put a fake joint inside someone, so in this case, that's the cup of the hip that's been replaced, here's the neck of the hip and the ball and socket that's been replaced, you can do computer work to generate where all the friction and wear and tear happens. So this is not so sexy to show to you without a lot of videos and talk, but you can work out where wear will be happening more and change your prosthesis. So they have to work with industry, with people who are producing these devices and selling them into the NHS. And some of the work was also using what's called osteochondral scaffold. So I mentioned you can take other structures, pig cartilage or pig bone, remove all the cells so the body doesn't develop an immune reaction to that, and put it into people and let it repopulate with human tissues. And they've been doing work evaluating when you replace a bit of bone and cartilage, people who've got holes in bone and cartilage, you can put a scaffold in there and see what happens. But all this work has to be done before we can do it in humans. So this is a slow process of understanding. You can't just jump into humans and say, right, we'll shove this thing in you and see how it works. So there's an awful lot of stuff done behind the scenes before we even get to developing. One of the big parts of the work out of a couple of these work streams is developing the technological and experimental methods that become the standards that all companies have to use. And our bioengineers have been involved in a couple of new standards for implants and assessing wear and tear. So again, these are things that don't get high priority publications, but are important behind the scenes to make sure that hip replacements, when they go into you, are going to work for a longer period of time. And there's a lot of work here. Some of this has been led by Tony Redmond on the gait stuff, looking at understanding patient characteristics and then feeding that into the models that the bioengineers are using to predict survival of joint replacement. And these models, Tony has a gait lab in outpatients at Chapel Allerton, and they allow us to look at how people are using their bodies, and we've been using these for looking at hip and knee joints, and we can understand how they walk, how they take load, and then feed that back into the modelers who are devising joint replacements. Now I'm changing tack entirely to talk about drugs again. So lots of biomechanical bioengineering stuff. Then there's pharmacological interventions, which everybody's looking for this problem of osteoarthritis, this painful, stiff, function-losing problem of osteoarthritis. So what's come out just during the last BRC? This is just the last 4 or 5 years. So we've had a large randomized trial that we did, that we got funding for a few years before that. That study was completed showing that hydroxychloroquine, which is a common drug used for treating rheumatoid arthritis, we said, well, maybe that could be used to treat the inflammation we see in finger joints in osteoarthritis and reduce pain. That study was a definite negative. It was now a highly cited trial, and 2 other big studies followed this one and showed indeed no benefit for hydroxychloroquine in hand osteoarthritis. We spend a lot of time in the current BRC, you'll be aware, doing imaging work. Paul's shown the role of imaging in RA, where we're detecting early inflammatory arthritis, in ankylosing spondylitis, where we can show spinal disease that people weren't aware of. And in osteoarthritis, we've developed very sensitive artificial intelligence-derived measures of bone shape. And your bone shape is likely changing in your knees, for example, from your 20s and 30s. It's already starting to slowly change and flatten out. And at some time later on in life, you end up with this condition we call osteoarthritis. This was a study we did using an anti-bone drug. It's called a cathepsin K inhibitor, a drug that's used to treat osteoporosis. And we said, with this sensitive measure in knee osteoarthritis, can we reduce the amount of bone change, and will that help people's symptoms? And the answer is we were able to show with 2 doses of this drug using a sensitive measure that we developed that you could slow the amount of bone change, but it didn't help people's symptoms in a 6-month period. So that's not very useful. Just slowing something on an MRI is not going to mean much. And sadly, it means nothing to regulators who are never going to license that drug for treatment until it helps people's symptoms. One of the most interesting things we've been involved with was an analysis of a big company trial that used an anti-interleukin-1. Paul's talked about the tumor necrosis factors inhibitors. These are the small molecules produced, inflammatory molecules that, by the way, for those with rheumatoid arthritis, make you feel rubbish. When you've got active rheumatoid arthritis, your inflamed joints produce chemicals such as TNF and interleukin-1. And this is a therapy called anti-interleukin-1 that was used in a massive trial of 10,000 people with heart disease. So it was not an osteoarthritis trial. But what we noticed in the side effects of this trial was that the people who got the active drug, the anti-interleukin-1 versus placebo, had about 40% less joint replacements after 3.5 years. So that was telling us, hold on, this drug must be doing something to help people's pain. And we couldn't understand because the previous anti-IL-1 trials had never shown anything. But what we think this is that the people, and by the way, this just shows if you said you had osteoarthritis at baseline, the same effect was there. But what we think this study is showing us is that for the first time, we can pick a group of people who might get more pain, and that's a group who have an elevated systemic marker of inflammation. And nobody's ever seen that before. So all the people who went into this study had cardiac disease, and they had an elevated, highly sensitive CRP blood test. And we now think that that's going to be an important marker for who we treat with osteoarthritis. So I'll link that to another piece of work that we've done out of our osteoarthritis work streams in the current BRC, which was a large randomized controlled trial of methotrexate. Again, many of you will have heard of methotrexate, the commonest drug used around the world for treating inflammatory arthritis like rheumatoid, psoriatic arthritis. And we said, can we treat inflammation and help the pain of osteoarthritis? And what this large randomized trial did was show that we could indeed significantly reduce the pain of osteoarthritis after 6 months with treatment with methotrexate. But what I haven't shown on this slide, in a sub-analysis we've done, having seen the previous big trial data, we looked at all the people who had an elevated, highly sensitive CRP at baseline and found that the pain benefits were much greater in that group. So for the first time, we've got a clue on a marker for something that might help better pain response to drugs in osteoarthritis. So that underpins where we're going to take a whole lot of our work in the next BRC, will be to look at that group. So I'm now going to finish up talking about, there's a whole lot more of people who did well out of the BRC, students who developed careers, advanced researchers who moved on in their careers through the BRC. There's an awful lot of work and productivity and output that's come. And I think this BRC, the existing BRC, really earned its keep, because for what we got from the government, which was about 6.7 million over 5 years, we produced a huge amount of stuff over a long period of time. And we shouldn't forget, as we move on to the new BRC, Paul Emery, who led the previous iterations of this BRC, is clearly a renowned world leader in his area, and also the established person who helped us get the first BRC here at Leeds. So he grew the critical mass of people that he now showed in the slide, and I've added to that with the sort of osteoarthritis and bioengineering side. And that's what you find over time, that research groups are about people. They're about having the right cadre of people, and having the cadre of people who've got some expertise across a range of areas. And we've managed to maintain that over a long period of time. So my thanks to Paul for all the work over a long period of time that culminated, first of all, in that first BRC, and then in us being successful in successive elements. We then realised over the last few years that the Leeds was bigger than just musculoskeletal medicine. There was a lot of other good research going on in Leeds, and we wanted to try and get that recognised. And many of us think we probably could have done better last time in the previous round had we gone for more disease areas in our BRC. So what's the new BRC? So this has just been awarded and started on the 1st of December, a few days ago. The goal of our BRC is it will be very patient-centric, patient-led, that we try to increase the speed at which research is adopted. Currently, if you have a good idea, it takes you about 17 to 20 years to get that idea through to the clinic. So my methotrexate knee trial, we started doing the first pilot studies for that some 13 years ago. And it still needs another big trial before it will change practice. And we're looking at how we do that. So it's very slow. People have ideas, you have to do a trial, you have to populate that, you have to publicize that trial, but to change practice takes forever. And we've got to really look at how we do that. We will be collaborative. And I think what you've seen from Paul and I is there's a wide range of clinicians, researchers, scientists, bioengineers, and patients in everything we've done. We've changed that culture. That's now standard. We've now got to bring in even more specialists, and I'll come to that in a minute. Equality, diversity, and inclusivity is just taken for granted. That's the color in the background of everything we do. And we, of course, want to grow our staff. We want to grow our young staff. Paul showed a couple of examples of people we've kept. We haven't always been able to keep all the people we wanted to because there was no jobs. And the BRCs don't have money that keep people going. This is an infrastructure grant. It's supposed to prime people to go off and get other grants. That's the point of a BRC. But we want to make sure the good people come in and we help support them. And the Leeds reputation for doing that has been spectacular, especially under Anne-Marie Keenan, who many of you will know, who really has really revolutionised how we're supporting and keeping our staff through, especially for the non-medical staff who are often historically undervalued, you know, in areas that are not medically driven. So we want to keep these things in our strategy. This is sort of our background colouring of what we do. what areas? I've got some things covering the top of my slide. I hope you can see that okay. So you'll see that there are now 6 themes in our new BRC. We were a single theme musculoskeletal. Now we're 6 themes. And you see musculoskeletal is still the biggest because we've got the track record, we've got all the experts we mentioned. But as well now, we've added in surgical technologies, cardiometabolic disease, that's the links between cardiac and diabetes medicine, hematology in collaboration with the University of York. For the first time, we're bringing in other universities in our region in, and NIHR asked us to do this. They really do want an interlocking network of research around the whole country. They've made it clear that they want research done where the patients are, not just in Oxbridge, that the research should be everywhere around the country where the patients are, and that's going to mean bringing in groups everywhere. We've got pathology and we've got antimicrobial resistance. Now, just to say to you, bugs becoming resistant to antibiotics could well very soon, if not already, become the biggest killer of people around the world, even more than cancer. So antimicrobial resistance is a big problem, developing new therapies and using what we've got well. And you can see there that underpinning all these groups will be some of the things you're familiar with. PPIEP, which underpins everything we do. We've got to grow our data, digital and AI work because we are still very basic and we're still behind other places in the country. Just linking patient records, linking stuff across trust and uni, allowing researchers to have access to anonymized patient data. All these things are major, major headaches that's going to take a lot of work. We know we're good at doing early trial design and we've got a clinical trials unit that can help us with a lot of that. And we're committed to a positive research culture, as I mentioned before. So we've gone from being just an MSK BRC to 6 different disease areas. Oh, and some of those areas, those arrows, by the way, going everywhere on this graph, show some of these people already collaborating. So the osteoarthritis team work with orthopedics quite a lot. And increasingly with this metabolic osteoarthritis syndrome I mentioned, we'll work more with the cardiometabolic people. Paul's been working with the antimicrobial people because we know that chronic gum infections are part of the link to people who develop those autoantibodies for rheumatoid arthritis. So there's already quite a lot of cross-linking between some of these themes. And this is just to give you a little more about, it's not really possible without a couple of hours to talk about what each of these themes will be involved with. And Amy, I've no doubt over time, as we develop our new PPIEP groups, that we will be able to do some presentations from each of the themes that talk about what they're doing and what their areas. But I think you'll have gathered from what Paul and I have said that we will continue to be working in both inflammatory arthritis and mechanical joint problems, that both are really important areas for the MSK. Cardiometabolic, they've got big imaging interests like our own, and they've done a lot of work about using imaging to detect early cardiovascular disease. And now they're looking at areas for heart failure, especially in the diabetic patients. The surgical technologies bit is looking at how to monitor patients remotely. There's really interesting technologies now where you can put sensors inside joint replacements and people will be walking around transmitting data. Watch out when you go through the airport thing now, it's going to be a different thing altogether. But these will be able to tell us, you know, have we increased this person's steps? Have we increased their activity? What sort of wear and tear are they taking through their joints? So there's a lot of work going on about sensors, both to stick on the skin and things implanted within joints. And then there's a lot of work about using robotics and other things to do operations more sensitively. And David Jane has done a lot of work around using robotics and robotic endoscopic work to how to understand how we treat bowel cancers, for example, better. So a lot of work going on in that area. In pathology, we're using artificial intelligence. They're taking a biopsy from somebody. It goes on to this massive whole room screen where they can do lots of fancy analysis to understand different characteristics of the cancer. Is this going to be more malignant than this cancer? Not only have we cleared enough of the cancer, but what do we understand about the type of cancer this is? Is this a group who we should be giving more therapy to immediately or not? So they're doing some really good work, and they've already got big NHS grants on improving accuracy in diagnosis in the lab after you've had a biopsy. I mentioned antimicrobial resistance. We'll be looking at, they've got some existing models of working out how new drugs will work in people, and they've lots of companies coming now to use these laboratory models to work out how their drugs are going to work, but also how to find certain problems that there's no treatments for at present. And some of the big issues are people who get cleft difficile problems after having antibiotics for too long. They develop an overgrowth of bowel bacteria that is very resistant to treatment. So they're doing some work to reduce that. So again, heaps of work. And in hematology, it's going to be a lot about the speed of diagnosis and improving the treatments for certain blood-related cancers.
So you'll have noticed that we've grown a P in patient and public involvement and engagement, and that additional P that we've grown is for participation. And I think our interpretation of participation is how we use patient and public involvement and engagement to make participation in our research access activity more inclusive. And I think that leads nicely on to the 6 objectives of our patient and public involvement, engagement and participation, so PPIEP strategy. So the first one is PPIEP is representative of underserved communities. And you might have heard this terminology used quite a bit in the last year or two, particularly by colleagues working for the National Institute for Health and Social Care Research, that's the NIHR. And the reason for this is, in the same way groups have been spoken about as underserved by health and social care services, the NIHR has adopted this train of thought as well, whereby groups are underserved by research. And the reason that they're underserved by research is activities and participation opportunities aren't designed in a way that's inclusive for their needs and their accessibility. So we've taken, like I say, we've taken this interpretation of participation and linked it in with sort of our representation of different groups within research, and also how we make those opportunities inclusive. The second, to work as one NIHR. So Philip mentioned that as a really important factor about how we work across the region, how we work wider with our different NIHR colleagues, and particularly in Leeds, where we have a lot of different infrastructure organisations, such as the Clinical Research Facility, the In Vitro Diagnostic Cooperative, and the Surgical MedTech Cooperative. In the same way our research teams will work together, we'll also work together as a PPIEP team as well, so sharing resources, and also we'll work beyond the NIHR and think about how we involve our colleagues from the University of Leeds and Leeds Teaching Hospitals Trust. And I think a step in that direction is the recruitment of a patient public involvement and engagement manager for research and innovation at Leeds Teaching Hospitals Trust. So that's Hanif Ismail, who some of you will have met. He's been in this presentation as well. So it's really good that we're actually starting to see a growth of PPIEP staff as well. The next one, to create a culture of public partnership in everything we do. Again, adopting NIHR terminology when it comes to PPIEP in public partnerships. And the reason that terminology is so important because it talks about how we work in partnership with people. So very much around the principles of co-production, that everybody's got an equal seat around that table, and the opportunity to participate around that table is equitable, meaning everybody can participate equally. Increasing awareness and engagement in research activity through effective partnership working. So I've already talked about how we'll work with one NIHR in Leeds, and also how we'll involve colleagues from the university and Leeds Teaching Hospitals Trust. But we'll actually go further than this as well, and we'll look to have a member of our team based in an organisation called Healthwatch Leeds, and this really embeds our PPIEP within the community and allows for those relationships that are really important to work with different groups to grow in the place that's accessible for them and where's comfortable for them, as opposed to it all being based in a hospital. To recognise the contribution of value of public contributors. So there's the monetary side of this, of course, there is in terms of recognition payments, and we will endeavour to continue putting recognition payments and expenses into the grants that we have moving forward, and that's very important and something that we do. But I think there's the other side of things as well as impact, and impact is one of those really neglected areas, unfortunately, and something that we really need to improve in the future is about how we share impact of activity and outputs of activity with our patient and public partners from the activity that they've taken part in. And then finally, to plan, monitor and evaluate impact. So I suppose that just leads on perfectly from what I was saying, to make sure that we really capture that impact, and we can sort of say, well, we did this and this is the impact of it, so we can show how important PPIEP is in the research that we deliver. And our underpinning values for this. So to be committed to equality, diversity and inclusive practices, to respect, to have respect, integrity, to manage confidentiality, to be transparent and accountable for our actions as well. And then